recording. Still getting it. So make sure that first and foremost that all of this extra business does in fact feel good and comfortable for you. So that you know where you need to be and when, and that by next week at the very least it starts to feel pretty comfortable. And if it doesn't, and that you're not confident that you're going to the right place at the right time, that you contact your instructor or the appropriate TAs or whoever it might be sometime today or over the course of this weekend. So when you come back on Monday, that you can start to come back with a little more comfort and confidence with where it is that you need to be going and what needs to be happening. And I know that it is difficult, but make sure that you certainly at the very least feel comfortable reaching out to someone that can make you feel at least a little bit better about what's going on. Looks like most of you are, at least for this class, getting there, and I know it's very frustrating. So I want you to know that you're doing a really great job. So there's that. For class, make sure we have several assignments that are closing. So make sure that you're doing them. For lecture, we have our extra credit syllabus quiz to do. So that actually closes blocks. Right, so you won't be able to access it tonight at midnight. So make sure if you haven't done that yet, it will, remember this is the way that you can practice with Proctorio. I know there have been some trouble with the lab Proctorios, mostly because the lab had a nervous breakdown and had a breakup. My lab is actually working. I don't know if Dr. J's is yet or not. She had a lot of trouble and IT got involved. So, but all of my stuff, if you have me, is working. And the syllabus quiz was never broken. I guess it's just a little sweeter around the edges. So if you are still having trouble with that, that's okay. But make sure that you are working on that today. If you are having trouble, I'm actually free after this class. So we can do an MS Teams meeting. And you can actually share your screen. So I can see what you see, and we can work through what your problems are and get you right on track. And I've had a couple of students do it, and I usually get you all fixed up in less than 10 minutes, and you can go about your Friday and not your professor in the face and get a break from me, but also do it in a way that's frustration-free. And then get those extra credit points. The quiz itself is super fast. It's 3 questions. So it's really more about getting you going with Proctorio than it is about the quiz itself. Also, don't forget that the Learning eLearn module is due closing, locking on Sunday. Okay, so this is a little bit longer. The idea is that it's teaching you all the different ins and outs of using eLearn for those of you who have not had the opportunity to use it before. So it's going to teach you to get in and out of the gradebook, to find your calendar and due dates and all of that business. So make sure that you start that before the 10 minutes that it's due on Sunday. This doesn't have Proctorio in it, so you shouldn't experience any problems when you're doing it. Same sort of issue though, if you are having trouble or if eLearn is frustrating you, definitely just reach out, because this is the week to get those kinks and bumps out, right? Because we're going to really be hitting the ground running moving forward from here. Okay. All right. So on Wednesday, we were down. Can you do me a favor and just half watch that this thing records the whole time? Yeah. It just like randomly quit on me at 9 a.m. and I'm real shook about it. Yeah. Okay. On Wednesday, we were just finishing up the 3 houses that we were on. So we just finished up cell theory and all the things that it takes to be a cell. So our very last house, oh my, our very last house is genetics, all right? And so this feeds in nicely as we were talking about DNA, okay, let's put it here, is one of the major components that you need to be a cell. And we know that being a cell is something that is a core component of being alive. So we can start to see all of the sort of catwalks or bridges between our houses. So we really want to focus on what is it that is a key thing about genetics or DNA. Okay, so that's our last house. Yeah. Okay, so let's look at this. So most of the time when we talk about DNA, we're going to talk about it in the form of chromosomes. Okay, so chromosomes are just packaged units of DNA. All right, so if we think of DNA as all of the stuff that's in your house that allows you to be you, do you, and live your extra life, okay, when you go to move around, right, so like when you moved into your dorm rooms, how did you move your stuff around? Did you carry it one by one? No, man, no one got time for that. What did you do? You put in boxes, right? So that's the same thing that DNA does. It puts its stuff in like boxes. And that's all chromosomes are. Boxes of DNA. Just like you put your things that make you into boxes, DNA puts its things into boxes to make it usable or movable. And that's an analogy I'll use a lot this semester. Okay? Okay. So, DNA itself, right, is the substance of genes. And we're going to talk about genes as the things that code for something. You have a gene for hair color, a gene for height. Okay, so this is the term for the functional units of DNA. Okay, so when we put all this together, then chromosomes are these subunits that you can then inherit. Right? This is how we pass things around. Right? You put things in a box to give it to somebody. And they ultimately then control, right, so they have the genes on them, pretty much everything about a cell or an organism, at least partly. Right? Not everything about you is exclusively controlled by genes. Okay? So development, your looks, your actions, all of these are in part controlled by your genetics. Now, it's not exclusive, right? We know height, for example, is genetic, but you can do things with your actions that change height, right? If you work out, if you do steroids, these can have positive or negative effects on how tall you'll grow, regardless of what your genes say, right? Okay? So, at least in part, your genes will have an effect on all of these different parts of a cell or an organism. Okay. We'll talk about exactly how these relationships affect these outcomes much later in the semester. Okay, but for now, just knowing that they do is where we want to start. Hi, now that's our last house. So all of that feel good? Any questions? Okay. So what we're going to do next then is bounce into the second sort of half is probably not the right word, but the second part of our themes unit and look at how can we take some pieces and directly apply them to science. And a lot of the discussions that we're going to have here will be really helpful to the lab that you're going to have next week, which is going to be running a mini experiment in your labs. So we're going to talk about all the different pieces that you'll need to do that successfully. And we're going to start with the two different types of thinking or reasoning, which are called inductive and deductive, okay, both of which you have probably already used and certainly been exposed to. Okay, the first type of reasoning is called deductive. Okay, and this is the one that will probably feel most intuitive and you've probably seen the most on TV shows. Okay. So I like to call this the Sherlock Holmes or the CSI thinking. Okay. So deductive reasoning is funneled reasoning. So in this case, we're going to take lots and lots and lots of pieces of information. So that's going to go at the top of the funnel. And use them to funnel down, okay, to all come down to one point or one small conclusion, one specific conclusion. Okay, so this is where the sort of CSI Sherlock Holmes thing comes in. Okay, so if we think about a classic CSI episode, right, you have the bloody glove, the knife, the shoe print, and Jerry's lack of alibi that all points to him as the murderer of Cindy. Okay, so that's where our funnel comes in, right? The shoe print, the bloody glove, the knife, all of that stuff is our information. Okay, we use all of that to funnel down to point to one specific conclusion. Jerry murdered Cindy. Okay, we can't use that information anywhere else. Okay, the shoe print was at one murder scene, right? Okay, the glove only fits Jerry, okay, not Carl. So we can't use this to apply to any other murder scenes, to help any other murder victims, to pin any other murderers, right? Okay, so this only helps us for one case. Okay, every time Sherlock Holmes gets a new case, he gets a new book, right? A whole new start. Every time we have a new case, it's a new CSI episode. Okay, it's a different funnel. So this is why it's a downward funnel. We have one small conclusion. So this is deductive reasoning. Okay, Sherlock Holmes deduces. If you've read the book, he actually uses that phrase. I don't know that the new show actually uses that language, though, so you might have to watch the old school stuff to get it. Okay, that feel good? Okay, let's compare that to inductive. Okay. Inductive is more commonly used in scientific applications, so you might see it in some science shows, and you've probably used it yourself. Okay. So inductive is my inverted funnel. Okay, so down here at the bottom of the funnel, we have our information. Okay, now, we still might have a lot of information. Okay, like we did before, we had several pieces. And ideally here, we would have more than just maybe 3 or 4 pieces of information. Okay, but our information goes to the bottom. At the top, okay, instead of a small conclusion, we have a big or a broad conclusion we want to make. Okay, so if we think about the image we have here, if I'm looking at a lake that is polluted, I might gather a lot of information about that lake. Okay, what are all the pollution parameters? Okay, what are the organisms that are living there? How big is the lake? Blah, 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 right? So that's all of my information. Okay, when I'm done, though, I don't want to just make a conclusion about this lake. Okay, I want to try making a conclusion about all lakes. Okay, if I find another lake that is polluted, what organisms will I expect to find there? Should people be able to drink from there? Maybe I can make some policy decisions, right? And so that's the key here is we want to, here's our open funnel, and I want to try to make a conclusion that is bigger than the information that I have. Okay, and so this is what's really important here, is that I'm trying to say something important or big. Okay, not that finding the murderer isn't important. Okay, but I've only found one murderer. I'm trying to save more than one lake, for example. Okay, so this is why it's important that maybe we have more than one piece of information. Okay, but the goal is still, whatever conclusion you're making is going to be bigger than the information that you have. That makes sense? Okay, any questions about those? Okay, let's practice it. I like to practice. All right, here's our first practice. Let's get this out of here so you can see. All men are mortal. Harold is a man, therefore Harold is mortal. All right, so go into your mentee.com, okay, whether it's the website or the app. Okay, and remember when you go in, it's going to ask you for a code. Here's your code. 56 56 73 8. Okay, and tell me whether you think this statement is using deductive or inductive reasoning. Okay, let's take a look at this. Let's tackle this like I would tackle an exam question. Okay, so first I want to find my information. All right, so what part of this question is information? I have all men are mortal, and Harold is a man. Okay, so this first part is my information portion. Wow, that's definitely the word men. Okay, so now I need to decide my conclusion. So that leaves this part as conclusion, right? Harold is mortal. So I need to decide if I'm going to be drawing a regular funnel or an inverted funnel. So I need to decide whether this statement is a big, broad statement that I can apply to many things, or a teeny, small statement that only applies to one specific incident. So what do I think? What does this seem like? Is this a teeny small or a big broad? Why is this a teeny small? Because it's only Harold. Okay, so that's what's really key here. And it can be difficult to pick out, but in this case, this statement is only applying to Harold. Okay, I've been very specific. Okay, some of the information is more broad, right? All men are mortal. That's a big, broad piece of information. But the conclusion I've made is very specific. Okay, Harold is mortal. It'd sort of be like saying Harold is the murderer. Okay, it's only good for one person. It's very specific. Yep, that's kind of a word. Okay, so spelling aside, if I'm making a specific statement, does that have to be deductive or inductive? Deductive, good. Okay, does everybody see how I built that up? Now let's try another one. Okay, this should have automatically updated on your mentee, so you should not have to put in the code again. So Harold is a grandfather. Harold is bald. All grandfathers are bald. Okay, let's pick this one apart again. So, information. I'm going to use grandpa because it's easier. Okay, so we made some observations. Okay, we have a bald grandfather. So there's my conclusion. And they won't all be 2 and 1. And this one has a word count cutoff, so somewhat limited in how I could construct these. All right, so there's our conclusion. Grandfathers are bald. Is that a big, broad, open-ended conclusion or a tiny, specific conclusion? It's big, broad, big and broad, right? All right, I'm trying to make a conclusion about all grandfathers, okay? That's more than one, okay? Many individuals. So I'm going to have open-ended. All grandpas. Okay, so whether you guys just assumed I'd pick the other one or whether we're feeling very confident about it, right, we're all in the right place. Okay, but something else has also happened here. Okay, what happened with this statement? Are all grandpas bald? No, right? And so this is an innate potential issue with inductive reasoning. Because we're trying to make these big, broad statements, we're more likely to be incorrect. Okay? And so this is why it's really important that we get lots and lots of information. And of course, in this case, we definitely did not, right? In this case, we have pretty much one observation on one person. So this would be like a toddler trying to understand what makes a grandfather by looking at our own grandfather and understanding that this is grandpa. So certainly this is problematic. And so in science, we work really hard to not do the thing. Okay, so we have high sample sizes. Okay, there's a minimum number of sample sizes that you have to try to make sure this doesn't happen. And then we repeat our experiments with other people, right? So someone else does it, pulling from a different pool of samples, so you don't get the exact same result and see if you get the same conclusion. Okay? To help prevent sort of weird all grandpas are bald situations from occurring. Okay? Because there is such a potential from looking at just a few things and saying, well, I guess everybody's bald. Okay, great job. Any questions about that? Good online? Great. Okay. Next up is looking at how we can start to apply this thinking to the scientific method. Now, I'm not going to spend a ton of time on the scientific method here. One, because it's probably more intuitive out of all the things that we're going to talk about. And two, because when you're not doing it, it's kind of not that interesting to talk about. And you're going to spend a lot of time actually doing the scientific method next week in lab, and I think it's going to be a lot more interesting to actually go through the process of the scientific method and learning it that way than hearing me talk about it here. So we're going to do scientific method light. Now, one way or another, you guys have probably all seen the scientific method, whether it's literally meme-worthy or you've gone through it in some kind of science class in the past. So there's just a few places that I want to talk about it here, and then I'm going to pick a few spots in lecture to really focus on as we think about what makes a good scientific experiment, so you can apply that well when you're doing your experiments in lab next week, which is what you're going to focus on, designing your own mini experiments. So, let's get off the hot pink. When you're going through this process, okay, these first three steps, that is terrible, are very linked. Okay? So whether you're given your main question or whether you found a question to ask, okay, that's really where you're starting. Okay, and everyone kind of forgets this step. Okay, but when I ask people to tell me what the definition of a hypothesis, most people sort of give me the very colloquial educated guess business, right? Okay. Now, we're going to do sort of a more thorough version of what that definition is here later, but the key here is it's an educated guess, right? And so we're going to do the educated part of that, right? Which means that you're going in with an informed idea of what is going on. So when we construct our hypothesis, if then because, right, this is what I think is going to happen, and why do you think that is going to happen? Okay, that why needs to come from somewhere. And most of the time, right, you need to look up what's going on. What's happening here? That's very interesting. Okay. Now, particularly at this stage, I'm not expecting you to look up 20 peer-reviewed journals. Right? A little bit of a Google search is really more than appropriate. But most of the time, that's going to give you plenty of information to go into something with some kind of footing and say, okay, I kind of get it. This kind of makes sense. Right? And then have an informed opinion of what you think is going to happen and why. Okay. We're going to spend a lot of time talking about how to test an experiment. Okay, the idea of creating something that is testable and falsifiable. So then the last thing I really want to talk about here is this last step. Okay, one, when you're done, you need to reflect back and say, well, was I right or was I wrong and why? Right, so what happened? Okay, you actually do something with that, right? I know particularly in school it seems like, oh, I gave my teacher my paper and I got a grade and okay, it's burned in the fire pit at the end of the semester so I never have to think about it again, or I gave it to my mom and my mom still has stuff from high school for me, so whatever parents do inappropriately with things, it seems like, I don't know, way too long, right? So, but in the real world, right, these things are dispersed, right? We publish them and other people really do read them, right? We go to conferences and tell people about them. If you're really lucky, you end up on like the BBC, right? Things like that. So there is dispersal of information. And then whether you're right or wrong, you just keep going, right? You don't do one project. You're like, well, now I know everything about the way corn grows, right? So we do need to keep going. We can get better, more efficient, right or wrong. Okay? It takes many renditions of a topic. Right? And then the world changes and you almost need to start all over again. Okay, so let's zoom in a little bit, particularly on this hypothesis and experimental testing section, because that's what we're really going to spend the most time on next week, and that's why you're doing the checks lab right now to work on hypothesis building, and then we're going to work on building an exercise of an experiment in lab. So we're going to really focus on these parts of an experiment right now. Okay. As we look at things that we can do, the first thing we want to talk about is what is a hypothesis and what is even an appropriate question to ask scientifically. Okay, so questions we can ask. In the end, you must be able to create a hypothesis that is testable, okay, it's something that you can feasibly do. I don't necessarily mean like in a Bio 130 class, but a person could do, even if it's Elon Musk, right? A person could feasibly do, okay? And so it's testable and it's falsifiable, which means you must be able to say, what would this look like if I'm right? And that's always the easy question, right? I'm right. But also, what would it look like if I'm wrong? And sometimes this is harder. So you have to be able to look at your hypothesis and say, what does the inverse of this look like? Okay, is there one? And that kind of means you need to give yourself a little bit of pause. Is there an inverse? Okay, and that's a really important question to say, have I created a good scientific question and experiment? Okay, so let's practice some of these. All right, so we've hopped back into Mentee. This does have a new code now. So I want to ask, can I test scientifically that plants grow taller with fertilizer than without? So ask yourself those two questions. Okay, is it feasibly testable and is it falsifiable? Okay, that's a lot of yeses. So if I were to set this up as an experiment, what would my experiment look like? What do I need? How would I test this? Okay, so I need 2 plots, one with fertilizer, one with no fertilizer. very realistic looking plants inside. Okay, that seems testable. I can measure plants. I can have plots with fertilizer and without. I'm on team, we can test that. Is it falsifiable, right? So if I'm right, what will it look like? The one with fertilizer will be taller or bigger in some way or another. Okay, that's easy. I like being right. What will it look like if I'm wrong? Okay, so there's two options, right? So the one without fertilizer could be taller, or they're the same, or there's no change between the two, right? So there's multiple ways I can be wrong. That stinks. Okay, but I can clearly state parameters under which that I will be wrong, and those are still measurable conditions, right? Does everybody see that? Okay, so given all of that, this is a good scientific question. A little simple, but it's a good scientific question, right? We can clearly see all of the parameters. I can clearly test it and clearly see the instances where I am right and I am wrong. So it's falsifiable. That makes sense to everybody? Okay, let's try another one. Really? That's what you're trying to do to me? Thank you. All right. Can I test that flying space potatoes do not exist? Okay, and again, think through clearly the parameters. Okay. Now again, we're not weighted down by money, so don't let the fact that it's just in space be the issue. I work for Elon Musk. I'm very lucky. But cars into space, not a problem. Okay, so money's not the issue. Let's say time's not even the issue. Okay. So, but can it be done? And can I falsify it? Let these be the issues. This one's not as straightforward, right? Okay, so let's take a look at this. If I want to test that flying space potatoes do not exist, what do I have to do? I get to go to space. That doesn't sound too bad. But then what? I have to look through all of space and not find a space potato. Okay, that sounds kind of problematic, right? Now, on the surface, I feel like you have to take all of space, which is infinite and expanding, set it up into a grid system, and then explore each quadrant, and then not find a potato in it. Does it sound like something that you can do? It could take a very long, infinite, technically, amount of time, right, if space is infinite. So herein sort of lies the problem. If you're tasked with exploring an infinite amount of space, we've gone like Star Trek on this now, right? But their goal is not to just explore space for the sake of exploring space. They now have to explore every inch of space to find one thing, or not to find one thing. Okay, so that sounds probably not doable. Okay. Now, is it falsifiable? So what would happen if I was wrong? I would find a space potato. Okay. Now that's almost the best version of the story because I can stop looking once I find a space potato, which is great. Now what about the other version of this? 8, 8, 8, 8. What if I wanted to say test that flying space potatoes do exist? Okay, is that testable? Still kind of has that maybe issue on it. But what's the falsifiable thing on that? That's that they don't exist. And now we're back to that's really hard, because if my point was to prove that they do exist, is never finding one proving that they don't exist? Not necessarily, right? So that doesn't even really have a falsifiable side to it. It just might mean that I didn't happen to find one. So this is a really problematic one. So we would say this is not a good scientific question. Right? We do not have clear parameters that allow us to successfully test and falsify this one. Okay, does that feel good? You feel good online? Nope. This. Okay. Let's continue to talk about our fertilizer question then, because that felt like a good experiment, so we can use it as the example moving forward then. So if I'm actually going to do our fertilizer experiment, then the next thing I want to do is make sure I set it up as what's called a controlled experiment. The point behind a controlled experiment is to make sure that everything else that can affect, in this case, plant growth, or in any experiment that's going to affect the outcome, is literally under control. Okay, so if I think about my experiment, my fertilizer one, there's lots of other things besides fertilizer that affects plant growth, right? Sunlight, soil, water, competition, okay? I don't want any of these things getting in the way of my final outcome, right? Because that's just going to muddy the water of my results. It's going to take away my power. And I like power. So having a controlled experiment gives me power to say fertilizer affected plant growth, not any of that hoo-ha. Okay? So we want to keep control of all of this other business, keep it the same, get rid of it, whatever needs to be done. So when I'm done, I can say fertilizer. Fertilizer affects plant growth, or doesn't. That's appropriate, right? Okay, so here's how we do that. Okay. First of which, we're going to create a controlled environment, also called constant. Okay. This is controlling for all that hoo-ha we just talked about. Okay. Anything that could possibly affect plant growth that isn't what I'm interested in. Okay. So I want to make sure that I have the same pot size. Right. I don't want some plants to have more room to grow than others. I want to give them the same amount of water. I don't want to just water one plant and then the other plant dies mysteriously from dehydration. Okay? Because then I am going to be stuck saying, well, it could have grown because of fertilizer, or it could have grown because it got water and the other one slowly died. Okay? That doesn't sound very convincing in the end, no matter how convincing I may sound. Okay? I want to use the same plants. Okay? If I plant radishes in one and collard greens in the other, well, collard greens just naturally grow taller, right? So it isn't very convincing to say, well, fertilizer made the collard greens go taller than the radishes when collard greens are just already taller. Okay, I've lost power there. I want to use the same number of seeds. If I put 80 seeds in one pot, they're probably going to die. There's going to be Hunger Games in that pot. Okay, they want the same amount of sunlight, the same kind of soil. Okay, anything that you can keep the same, right? Keep your power here. That feel good? Still with me? The other thing you want to do then is have a control group. Now this is what you already told me. So remember on our design page, you said we had our fertilizer one and our no fertilizer one. Our no fertilizer one is the control group. Okay? So the control group is going to have all of this stuff. Okay? So is the treatment group, and that's what this is called. Okay? So the fertilizer basically has the +1 advantage. Okay, we're giving it the one thing that we're interested in this case, fertilizer. The control group has everything minus the one advantage. So this serves as your comparison. Our fertilizer plant grew this tall, we'll say 6 inches. The no fertilizer plant grew this tall, say 4 inches. So if that's the case, since everything else is the same, I can say my control group did not grow as tall as my fertilizer group, right? Fertilizer makes plants grow taller, in this case 2 inches tall. Okay, we'll pick up here and re-summarize on Monday. Remember, Fallon has a Socrative for you. Yep, they should get used to that. Okay. Make sure you fill out your Socrative. Remember the room number is Arnett 11. You'll go last name, comma, first name when it asks for yours. And when it asks for the teacher's question, you're going to put in the slide that we're on. And when you're all done, the back 2 rows you can leave. If you have any questions, make sure to get them asked. Remember, I said that I would be free after this class, so if you do need help, be sure to email or MS Teams me, and I can pretty much help you right away. Otherwise, have a great weekend. Try to get some relaxing in. You've all earned it. Make sure to spell your name. Oh. And you're in there. Go ahead and hit it. You should be able to.